Today we will discuss the NGS data analysis. So NGS data analysis, that's after that you see those sequencing data, and then you want to understand what does it mean. And as Adrian just mentioned, that you will see the one of the folder. It's called NGS data. Under that folder, I upload like 20, 30 files. Each file so basically each one is one sample, and each sample it contains hundreds, thousands, even millions of data in that file, and so it's been compressed. You can see the file name, the fastq.gz. So normally you will not be opening it. You cannot just click it and open the file. So that today we will just go through this process to see how we can read the file. Okay, to read the file, understand the file, and analyze the file. So basically, that the one you see in the blackboard, it's called, each one of them, it's each sample, and each sample contains hundreds, thousands of sequence. Those are the raw sequence, all right? The raw sequence that will not be able to see under the normal situation. Okay, it's not possible that you just click it and then you can see what's inside. All right, so you have to go through all this process, and then you will be able to see the detail. All right. So the first process that we need to do is to demultiplex the sequence. All right. And once you demultiply the sequence, you have a demultiplex the sequence, then you can join the pair together to make a one full sequence, and then you denoise, and from there, right? So today we will go through this process from here to here. And then from there, you have the representative sequence and the future table, and then from the representative sequence, you can do the taxonomy classification, and because I will also show you the process using those standard taxonomy and phylogeny protocol, and then to assign each sequence, right, to each microorganism. And then from the future table, then you can perform those abundance analysis and diversity analysis. So that's the main goal, that you have these NGS data and go through this process, and to give you this readable information that you can use for further analysis. Okay, so this is very simple of workflow, and for the chain tool that we will do. And of course, as I mentioned, that we will use this 2019.10, this version, although they have a newer version, but because they developed some new version, and also they disable some particular function. So I'm not very particularly interested in those newer versions, so I continue to use this version. And of course, then you can get all the help from the Chine website. Okay, there's docs.chine2.org, and then you can get all the help from there. But we will go through that today as well. So first of all, we have to understand the metadata. Okay, to process the sequence. To process this sequence, all right, from here to here, the first step. Before you do that, you need to have the metadata. And the metadata basically provide the key to gain the biological insight from the data. And that's usually specific to a given microbiome study. And collect the sample metadata is typically one of the first steps you would take before beginning the chain to analysis. Because in this chain to metadata, not only you assign the sample name and also correlate the sample file to the specific sample name. You also give some all the metrics, all other measure, all other data that you can put into the file. Okay, and normally it can be a TSV file or .txt. And the one that I sent you, oh, I did not attach that. I will attach that, okay, after the lecture, TXT file, okay? And the TXT file, then you can view this, and you can view this under the Excel, which is easy, and also you can edit the file easily, okay? That's a metadata, because from those trying to process, when you do the programming, and then they will all refer to these metadata, and so that they can process the analysis. And later on, you will see a lot of .qza file during the analysis. This is the trying to artifacts. That's the file produced through the trying to analysis. Okay, and this is a typical format of the metadata. So you can see that we have this the file path to the forward sequence, the file path to the reverse sequence, and also we have the index. That's the index that connect to the, during the NGS, you have to prepare a library, and then you connect to the sequence, right? And also you can give them a different condition or different location, so that's all information can be stored in this metadata, okay? So the sample metadata that include the technical detail, like a DNA barcode, right? That used for each sample in the multiplex sequencing run, or the discretionary sample, like here you have discretionary sample, and this is a barcode, right? And also we have the future metadata. That's the future annotation. So that's after you perform the taxonomy assignment, then you get the future data, metadata. So that each sample, they will assign the different taxonomy. That's the future data. Now we will see later, okay? And the demultiplex, yeah, that's the first step here, as I mentioned, demultiplex, right? The first step, the demultiplex. So that you can demultiplex single end or pair end sequence read, and then you can visualize the sequence quality information, okay? So for today's our sample, so that we have normally that you will see it's a MySeq, for example, MySeq reagent kit V3. So you can have the read length for 2 times 300 base pair and 600 cycle kit. That means this is a pair end, so you have 2 times, and each pair, each end is 300 base pair. So total is 600 base pair. So it's called 600 cycle. All right, and so that each read they can read these the 300, and so the total number read could be up to 25 million. All right, so that's the typical analysis that you can do. So here the single end, pair end sequence read. So the single read that run the sequence from one end of a fragment to the another. That's a single read. For pair end, so you from one end to another, and then started from another end back opposite side to the original end. So that's called pair end. And single read run are faster, cheaper, right? And sufficient for profiling, counting study like RNA-seq or ChIP-seq. So this is single end, but normally we perform the pair end. It's more accurate and more precise, and no mistake. So that's pair end that give additional positioning information in the genome, made a good choice for de novo genome assembly. and also easy to resolve structural rearrangement in the genome. That's the pair end. And relength. So relength, basically, the relength is just, so as I mentioned, the cycle, that's the number of base pair that you can read at one time. And the number of cycle correspond to the output relength, okay? So if it's 600, the cycle is 600, then the relength is 600, okay? And longer relengths give you more accurate information on the relative position of your bases in a genome. They are more expensive than the shorter one. And the 50 cycle, I mean, only read the 50 base pairs. They are typically sufficient for simple mapping of read to a reference genome. And the RNA-seq profiling, also a 50 cycle will be good enough. And number of reads. So during the sequencing reaction, sequence the base pair, all right, it's called, or reads are generated. Each sequencing platform and the instrument yield different number of reads, okay? And the depths of the coverage. So depth of coverage is so that basically mean a sequencing run generate reads that sample a genome randomly and independently. And these reads are not distributed equally across the entire genome. And some bases are covered by fewer reads and some by more reads than the average coverage. So that means the same position of the nucleotide can be read many times and many reads, or can be read fewer times and you have fewer reads. And so you need to have a depth of coverage. So that means if coverage is 100 times, that means each base was sequenced 100 times. Okay, the more frequently a base is sequenced, then the more reliable a base is called. All right, so that's called a depth of coverage. So that means how many times a sequence can be sequenced, a nucleotide can be sequenced. And this is some minimum coverage that required. So for example, DNA sequencing, the resequencing that normally you need to be read 30 to 80 times, the novel assembly, the 100 times, and chip sequence, the 10 to 40 time coverage is enough, okay? And the days of coverage for RNA, so you can see you have different coverage for the small genome, intermediate genome, large genome, and they have different the requirement, okay? Because the RNA, basically RNA, determine the amount of coverage needed for an RNA sequence experiment is difficult because different transcripts are expressed at different level. So that means more read will be captured for highly expressed gene because they been transcribed more. So they have more RNA. So then when you perform the sequencing, that particular one can be sequenced more frequently. So they have higher coverage. And for the less expressed, low expressed gene, then you have a lower coverage. So in general, for large genome, we recommend 25 to 30 million read for differential expression studies. Okay, so that you can cover everything. Okay, so that's the depth of the coverage. And this is just the process of the NGS sequencing. So basically, replicate are essential in any biological experiment. So for the sequencing, also we need to have the replication, and samples are subject to the variation that will make the biological replicate important to a statistical significance. Okay. And randomization. So randomization, basically, you can reduce the instrument effect, systemic bias, and the potential for the occurrence and the effect of the confounding factors. Okay. And also you have to be careful about a poor quality sequencing run. So when design the sequencing experiment, careful attention to the information is important, okay, so that you don't have a poor sequencing run. And during this process, you have to be careful about the PCR duplicate and the low quality read and those kind of situation. And also what you have to perform the library preparation, because you need to put these primer and index so that you can identify each sequence, and also where they belong to which sample. So that's the all those important factors that can affect your sequencing result. And for the NGS library preparation, so you have to choose a library preparation kit. You can do this, which this is being customized and also commercially available. Okay, and the library construction, basically, you prepare the nucleic acid target, right? And then the target, basically, you are pick a particular, based on the application, you pick a target, and then you have different primer, so you can duplicate the sequence and then perform the sequencing, right? And based on the different application, you have a different procedure to prepare the library, which is not what we want to discuss today. Okay. And of course, to prepare the library, you need to fragment the DNA first, and to fragment the DNA, so you have physical fragmentation, and you have enzymatic method or chemical fragmentation, different approach. And so you are to shear the DNA into small pieces, and so that it's easier for you to attach the primer, and also to attach an index sequence, so that you'll be able to identify the sample. Okay. And for the sample that I gave you today, all right, it has been sequenced, and so basically this we use the primer. It's called a 515 forward AO6 reverse primer, and this basically will amplify the variable V4 region, which I will discuss later. The V4 region, and then so that we'll be able to identify the microorganism sequence. So this is the first step. After you collect the genomic DNA, you fragment the DNA, then you perform the PCR reaction to amplify this particular target. And after the amplification, then you can perform the sequencing. Okay, so you collect the DNA, and then you have this PCR amplification, and this primer is a variable 4 region. After this PCR reaction, then you perform the sequencing and then data analysis. And today we focus on this step here. And so this is what they did for the library preparation. So you have the sequence that you collect, and then you add the linker primer sequence, all right, so that you can perform the PCR reaction. And then you add the barcode, so that you can identify which the sequence, which sample they belong to. And then you have adapter B. This is to perform the sequence in the different platform. All right. So this is first step. You have this primer sequence so that you can perform PCR. Then you add the barcode to identify, to label each fragment so that you know which sample they belong to. Then you attach the adapter so that you can perform the sequencing. All right. So that's a typical process. And then during the analysis, as I mentioned, we have to demultiplex. Right. The first step, demultiplex. Demultiplex basically, we use the computer program, the try to remove this sequence and remove this sequence. So we use the program to extract the sequence in here. All right, so that's called demultiplex. So we will try to retrieve the sequence in this region so that we can analyze the sequence. All right, so this is the procedure. So we have this. We get this the data. Then we determine the format. Then we import the data into the Chine tool. All right. And then we demultiplex. Once you demultiplex, then through the quality control, you have to do the denoise to remove those low quality sequence. And then you can also filter sequence. You can assign them a different group for different analysis. Then you perform the taxonomy and classification and build a phylogeny. All right. After you build a phylogeny, then you combine with another data. All right. And then you can perform the alpha test, beta test, diversity test, and all other correlation tests. That's the purpose. Okay. And here, just to let you understand, typically the sequence that you will see will be like this. And so that we need to use a Chine tool to remove this extra sequence, extract only the target sequence so that you can analyze the data. All right. And so as I mentioned that the primer. So as you know that for the bacteria, we use a 16S ribosomal RNA to identify their taxonomy as classification. All right, because different bacteria, they have different sequence in this particular region, in this variable sequence. These areas are the conserved region, and this V1 through V9 are variable region, which means from this variable region, you can identify the sequence variation and assign their taxonomy. And in our example here, we amplify the V4 region here, all right, so that we can perform the taxonomy classification to determine their classification, the phylogeny, all right, based on the sequence here. So as you can see here in the genome, we have 16S ribosomal RNA that give you these small subunit, all right? And then the large subunit, we have 23 and 5S, all right? That's 23, 5S. That's a large subunit. That's for translation. And so for the bacteria, we use this 16S. And this is detail about a different commercially available primer that they can amplify the region. So for example, this V1, V3 that can amplify V1 through V3, 510 base pair. That's for this NGS machine, Roche 454, which is pyrosequencing, all right? And V3, V4, this one, that's the MySeq, okay? And this V3 to V5, they amplify this region, the Roche 454, and this V4 to 52, right? Which is the one we use, right? No, actually, we use this one. We use a V4 292 base pair. We amplify from here 515 to 806, right? 515 to 806. We use this forward and this reverse to amplify this region, stereo 4, the very V4 region. And then we will, based on the V4 sequence variation, then we can assign the taxonomy and phylogenetic analysis. Okay, so this is just to show you different platform you can use. They develop different commercially available primer that you can use to amplify your target sequence and perform the sequencing. Any questions so far?
No, Professor.
Mm-hmm. Did you see the flash on my screen?
Yes.
Yes.
Let me stop it. Let me do it again. So this is the overview of the NGS process and the data. Okay. And then of course the main task after you get the NGS data is to analyze your data and then classify the taxonomy, build a phylogenetic tree, and then also perform some population analysis. And I believe the Professor Warney, he briefly mentioned about those different analyses. So those are main analysis, including this alpha diversity, which is analysis within a sample, beta diversity, which is the between samples evenness, the distribution of the taxa, richness, number of taxa, rarefaction curve. So basically that will be the alpha diversity versus the number of observation. So that test whether an environment has been sufficiently sequenced to observe all the taxa. So we will do all this analysis, okay? And also, then a more advanced, you will also do the PCA and PCOA analysis. And that would PCA analysis that to show you which factor contribute to the variation, all right, of the sample, okay? And also you can do this MMDS, non-metric multi-dimensional scaling analysis. So for the alpha diversity, so basically also it's called a species richness. You are going to analyze the sample, analyze the data within the taxa within the sample. So that says the number of the species that are present in the system. Okay. And alpha diversity is the diversity in the single ecosystem or single sample. So that's why the data is within the sample. And the simplest measure is the richness, which is richness, right? The number of species. And number of species, which is the OTU, is the operational taxonomy unit, right? If you get 500 read from one sample and then 100 read from another sample, then you will expect more species, right? More species in this second sample. And all right, so you will have 5 times as many than this one. That's the alpha diversity. A rarefaction, that's a random collection of sequence from the sample with a specified depth so that you will know at the what depth is enough to sequence the whole species. So they say a rarefaction with a depth of 75 reads per sample. So that means if you sequence exactly 75 reads from each sample, then that's enough to cover all the all the species in each sample. Okay, that's the alpha diversity. And beta diversity, also here, so alpha, so they say we have these three different habitat: the woodland habitat, the hedgerow habitat, open field habitat. So for the woodland habitat, let's say they identify at least 10 different species, and the hedgerow habitat, they identify at least 7 different species, right? And this one identifies 3 different species. So for the alpha diversity, then for this habitat, then you have 10, this habitat is 7, and this habitat is 3. So that's the alpha diversity. Then for beta diversity, okay, the beta diversity basically that's between the sample, okay, between the sample, between the different different ecosystem, and then so then for example in beta diversity, the woodland versus the hedgerow, right? So it's a 7, and then hedgerow versus the open field, it's 8, and then the woodland versus open field is a 13. That's between these two, right? The 13, and between these two is 8, and between these two is 7. So this kind of analysis, okay, that's called the beta diversity. Beta diversity. So this woodland versus hedgerow is 7. That's because these are duplicate. These are different ones. So these are 7. Okay, and these two, all right, so these are different, so the total is 8. All right, total is 8. And this one versus this one, they are different, all different, so it's a 13. So that's called beta diversity. Okay, so that's the two commonly used diversity. So alpha diversity is the diversity within the particular area of ecosystem, so that expressed by the number of species in that ecosystem. And then the beta is a comparison of diversity between the ecosystem. So measure the amount of species change between the ecosystem. So the amount of change. So that's why woodland versus the hedgerow, woodland versus hedgerow is 7, because these 5 and these 2 are different from these 2 ecosystems. So the diversity here is 7. Okay. And the alpha diversity, so also there are different index that you will need to know, like the Shana diversity index, which is quantitative measure of a community richness, right? The richness. And then the observed OTU, that's just qualitative measure of a community richness, the amount of a species. This is called phase phylogenetic diversity, so PD, which is a qualitative measure of community richness that incorporate the phylogenetic relationship between the features. So that's a phase PD. Evenness, that measure the variation in the abundance of individual per species within the community. So if the community with less variation in the relative abundance of species are considered to be more even than a community with more variation in relative abundance. That's evenness. So these are some terms that you need to know when we analyze the data. And so phase PD calculate the phases of phylogenetic diversity. It's a measure of biodiversity that incorporates the phylogenetic difference between species. So it's a measuring the evolutionary history between the taxa. And Shana index, that calculate richness and diversity. All right, that's the phase PD, and this is just for your reference, so you can have a look at how they develop the formula, how they determine the species diversity. Okay, the alpha diversity, beta, the Shiner index, the alpha diversity, how they perform the calculation. So this is for your reference that you can do this. All right. And so this is the website that we will go through today. Okay. So any question? Any questions so far?
No.
Very good. Very good. So first of all, that we will start this the process. First of all, I want to know if you can just download the Oracle VirtualBox and the Chine tool now. Can you download these two files now?
Professor, I've already downloaded both of them, but like.
Okay. Okay, good, very good. So.
Me too, Professor. I already downloaded.
You downloaded, very good. Okay, let me see. Let me see what I have so we can continue the file. And did you install the Oracle VirtualBox?
Yes.
I installed
Yes, I've done.
virtual.
Okay, so let me open the file. Just a second, let me open the file. VirtualBox. Okay, so VirtualBox, all right, you open this. Oh, it's not here because I installed it already. So you have to click the installation. So I want everyone to perform that, to install the VirtualBox. Okay, and once you've done that, so just use this Blackboard. Raise your hand. How many of you installed the VirtualBox already? Very good. Okay, everyone. That's good. That's good. And now, after you install the VirtualBox, you open the Chine tool like this. And of course, when you download is this file, then you have to extract this file, and then you open this, then you will see file here. Did you all see these two files?
Yes, I have it.
Good. Good. And then since you install the VirtualBox, before we proceed to do anything, I want you to click this VirtualBox now.
Were there two that we had to download or one that we had to download? Just 2019.10, right?
2019-10. Yeah, that's right. This is because I already installed the Chine tool here so that you will see. But now I'm going to teach you the process. So first of all, you open the VirtualBox. Once you open the VirtualBox, right, then you double click this file. Then once you click this file, it will automatically install into this virtual machine. Can you do this now?
Professor, I clicked it and it says import. Should I click import?
Mm-hmm. Yes. If you're done, you can raise the hand so that I know, okay? Once you import the trying to raise your hand so that I know. Why it takes so long? It should be very quick, huh?
I realized it didn't unzip mine, so this is why.
Oh,
I
okay.
downloaded like that day, but I didn't unzip it. So now I'm seeing that,
Oh, okay.
and so I'm trying to unzip the file currently.
All right. So whenever you done, then raise your hand so that I know it. Okay.
I can import
But
the file
okay,
from.
but I think okay. Oh, you can't.
Charlie, yes. What's your question? You cannot import.
Yeah, so when I click the Chime 2 core, it doesn't open directly into VirtualBox, but I can import it from VirtualBox, right? I can just click on that file. It's
You click
essentially
on
the
the
same
file, import the appliance.
Right, okay, that's what I'm gonna do. Thank you.
Okay. And you know this now I'm going to show you the basic technology. So you import this 2019-10, then of course in this virtual box, if your computer have a lot of the memory, not a memory, because memory you only use one at a time, so it's fine. If you have a lot of storage, then you can later on you can import the latest version in the same virtual box, because once now I import only the China 2 2019. Actually, you can import many here. All right. So later on, after the class, you can import even the latest 2021 version here, so that you can go back and forth to use different version. Okay. But because my C drive, uh, the size of my C drive is relatively small. It's only 100 gigabyte. So quickly it fill up. When I import this China to 2019.10, it use about 15 gigabyte of my storage. So that's why I only install one here. Okay. It should occupy 15 gigabyte. Okay. Since we are waiting, let me see what else I need to upload. Okay, so I have these NGS data. Oh, metadata, yeah. The metadata I forgot to give you. So I'm going to give you metadata. Metadata. I still see 2 people raise hand. How about the rest?
I'm getting an error message.
You try to import the appliance. Error message. So when you try to import, right? They say import appliance. Let me see.
Yes. And downloads and now.
So when you try to import, then they will show you the appliance setting. Is that right?
Yes.
So you see this message, and then did you see this base folder and the machine base folder?
Yes.
And is the same as mine?
No, it's not in program data. It's in users, and then Talia, which
That's
is my
your username, and then
yeah, and then VirtualBox VMS.
okay,
And then
and all the rest are the same as mine here.
let me just double check.
Except this folder, that's fine. That's fine. Different folder, that's fine.
Can you go up a little bit? Sorry. It looks like it's the same. 9, 10, 1, 5, 7, 1. Yeah, it's all the same.
And then you click import.
Oh, now it's working. Before I was
Oh,
getting
no, it's
an
working.
error.
That's good.
Yeah, thank you.
No problem. Please, when you're done, raise your hand so that I know.
Okay.
Don't lower your hand now, okay? Because I only see Tertia's hands up. Good. One more.
Did you see my message in the chat, Professor?
If you have problem, then I can just send the file to the. I can send the file to the content here. Okay.
Wait, say that once more. I'm so sorry.
I also upload this China2 file in the Blackboard. So anyone who need it, you can download directly. Okay, it's unzipped already. Nafia, are you doing okay, Nafia?
Yes, Professor. Try to import the.
Okay. Okay. Zhong,
Yes.
how's going?
Well, chime. I'm stuck on the chime. It's not opening, so I'm trying.
So you click this one, it doesn't open?
Right. I it just uninstalled again and then reinstalling.
Okay. Good. We have 4 people ready. Yeah.
Where can I find the unzip file again?
Which one you talking about, Adrian?
The 2019 point, I mean 2019.10.
Yeah, I uploaded. Did you see that under the content?
On the content on which day? I mean under NGS 578 NGS analysis, or
It's still uploading. Yeah, because the big file is still uploading. Yeah.
oh, it's still uploading right now. So you're
Right,
saying.
right. It's uploading to the platform now. Yeah. Just wait a little. Okay.
Oh, okay, hasn't. Oh, okay, no problem.
Okay, good. Good. Charlie, did you get it done?
I'm 50%, part 2 out of 3.
Oh, okay. All right.
So it's progress.
Very good. Good. So, okay, so good that most of you has done. Okay, so when you're done, all right, so what you need to do is you should see this chime tool on your virtual box, all right, and then to start a program, then you double click this. Then we'll show you like this here. All right, click. Did you click this?
Yes, Professor.
Good. Now we wait for the program to be initiated. And now, if you come to this, once you reach to this stage, raise your hand so that I know.
Dr. Shen, mine's showing an error, so should I just reinstall it then? It's saying failed to open the session.
Okay. Okay.
So I'm going to reinstall it then.
Okay.
Me too. I got the same problem, the error, but I figure out that I don't have enough space in my computer.
Oh, okay.
Yeah.
Yeah, I forgot. Okay, so let's turn this off. Turn this off. Just close this. All right. I forgot that you need to do some change here. So can click the machine. Click the machine. Click setting. All right. So close. You can close the chime tool and then go to the virtual box, and then click the machine. All right, and then setting. And then once you open the setting, okay, so you go to the system, and here, very important here, okay, so the base memory and the maximum memory, so you just change these to the maximum that you can have, okay, so because my RAM memory is 32 megabytes, so I can bring it to 24 megabyte here, right? So you just bring it to the maximum that you can have for your computer, right? Bring this to the maximum that you can have. And then click OK. And now you open the Chime tool. When you get to this step, and raise your hand, okay? Good. Tintin, how are you doing? Tintin?
Yes.
Which step you are now?
I cannot open it.
You cannot? But you install the Chime into the VirtualBox?
Yeah, but it failed to access the file.
But did you install this Chime 2 in the VirtualBox?
Yes.
And but you double click this, it will not open.
Yeah.
You can close the, you can just go to the machine and change the memory. You change the setting. You maximize the memory that you can have. Change this under the system.
Okay.
All right. And for those who already reached to this step, okay, you click this Chime 2. For those who reached to this step, you click Chime 2. Then they will show you this option. Then you type the password. The password is the same as these words here. QIIME2. Okay, so you type QIIME2 and then sign in. You can close this. And then that will bring you to this page. Once you reach this page, all right, then you can click this one. The first button on the top left corner, all right, terminal. If you go there, it's called terminal, and this one Q2 Studio, and this is Firefox, this is text editor, this is files, this is welcome to Ubuntu. So I want you to go to the terminal, click the terminal. Then I will open the terminal here. Once you open the terminal, then you will see this Chime 2 list. Okay, I will just give you some basic command. So LS is like a directory. Okay, so you click the LS and then hit the enter. That will show you the different folder. Under these Chime 2 these the working environment. All right, that's LS. And then what I need you to do is change the directory. All right, change the directory to desktop. Okay, see the desktop here? So they actually update. You can do it, remind me later. So you go to the change directory. It's CD. All right, CD. DESKTOP. All right, first of all, you do the list, LS. Then you see these different folders, and so you identify the desktop. So you change the directory to the desktop. So you type the CD desktop, and then hit the enter. So now you go to the desktop, and then you just click the list again. So what I need to do is when you click list, you should have nothing here, all right? But I just performed some analysis yesterday, so I have something here. So you click LS, you should have nothing there. Then I want you to make a directory. So it's mkdir, okay? Make a directory. So you can do 708, make a directory, okay? 708. So that will give you a folder called 708. And then you hit the enter. then I will give you the folder 708. Once you create the directory, then you raise your hand again, right? So that I know. Okay, once this is done, I already created 708, so I'm going to delete this, okay? You use the backspace to delete it. So what I want you to do now, okay, you go to the machine, go to the setting, machine setting, click. Not this one. Machine setting. Oh, actually, not here. So once this is done, go to the desktop. You click the files. Click the files. Click the files. Oh, go to the machine file, machine setting and general. So from the general, the share clipboard and drag and drive. So the default is disabled. So I want you to change it to bidirectional. Here, both should be directional. Okay. So go to the general, share the clipboard, drag and drop. Okay, both should be bidirectional. Change the setting to bidirectional. So when you are done, raise your hand. Okay, so these should be bidirectional and click OK. Once you are done, you go back to the file. This one here, you see the desktop, and then you double click the desktop. You should see the 708. All right.
I'm sorry. In the terminal, all we do is type in ls.
The terminal here, ls, to show you the list. And I want you to make the directory 708. So you will type mkdir, all right? Make directory. So we do 708, and then hit the enter. Then I will create the folder 708.
Okay, thanks.
Once you create a folder 708, then we go to this file. You click the files. Then you change the setting. You go to the general, advanced, and change this from disable to bidirectional. Once you change that, click OK, all right? And then you go to the desktop and 708. And 708 you open, all right? So this not supposed to be there, but I already upload here. So what you need to do is you open the 708, all right? Click 708, so the folder should be nothing here. Then what I want you to do is you go back to the Blackboard, you download the Blackboard. Go to this NGS data folder, all right? Then you download all the file, right? These are sequence file. Each one is from each sample, all right? Download to your hard drive. Then I want you to drag this, okay? Drop this and drag and drop here, okay? Once you download those NGS data, I want you to drag and drop here, all right? So first of all, you just drag and drop this H1 until H4, all right? Only this 8 file first. Just drag and drop this 8 file into the 708 folder.
Would it be possible if you showed the terminal screen? Thank
This
you.
is terminal.
Professor, I've downloaded these NGS data and they are in like the zip file. Like we'll drag the zip file and
No.
or do we have to extract?
So when you click, they say unzip. So each of these, it's a zip file. Yes.
Yes.
You just drag this zip file. So is the format like this? The format should be like this.
Yeah, these are like when
.gz.
I try to download them, they download as zip file. So like, do
Oh,
I have?
then you have to unzip it. Yeah.
Oh, okay.
You mean you download the whole folder and
Yeah, I
then
have
it's
downloaded the whole folder.
okay. Then you have to unzip the folder.
Okay.
I'm sorry, I'm still having trouble with the terminal. When I put in like the top part comes up, but after, yeah, the next green line, I can't get the CD desktop. When I type that in, it comes up as no such file or directory.
So when you type ls, did you see desktop here?
Yes.
Okay, so then you have to type cd space and then this has to be a capital desktop.
Oh.
And then hit enter.
Okay. Thank you.
You're welcome.
Please, Professor, I can't download the whole folder. Should I download file by file?
You download the whole folder, then you have to unzip it, yes.
Professor, I have dropped the
Yeah, drag
FASTQ
and
file
drop
of
the file.
H1 till H4, 8 files.
Yeah. Once you the drag and drop these A files, then let me see what else you need. And then you go to the Blackboard, then you should see the 708 meta.txt. So I want you to drag and drop the file here, all right, under the desktop. So here's desktop, okay? So the home, you have the desktop. You click desktop, you should see the 708 and nothing else. So you need to drop the 708 underscore meta.txt file into this folder. Once you've done this, then raise your hand, okay?
Please, Professor.
Yeah.
Please, Professor, I'm not sure. I download the file from H1 until H4, and I unzip it, and I can, but I'm not sure because I see in the screen it's Meta 708 Meta. I'm not sure about the last step you did.
Did you download the file individually?
Yes, I download
Oh,
the
okay.
file.
If you download the file individually, then you do not need to unzip it. You just drag and drop it.
Okay, I will try.
Oh, see, my screen way too long, so they will went to sleep. Okay, once it went to sleep, that's fine. You just click it. You just hit the enter again, then go back here. Then you just type try to, then they will resume. So basically, what we need to have is this A file under the folder 708, and then the 708 meta file under the desktop. So you should have one folder and one file so that we can begin, all right?
When I tried dragging into this in the 708 meta file, it didn't work for some reason.
It doesn't work?
No, it won't drag into it. It gives me
Okay,
like that X.
so let me show you. So normally what happened is, let's say I drag any file, all right? And then you drag it into your destination. Then you should see plus copy. If you see plus copy, then you can release your mouse so you will drag. And otherwise, if you see like this red sign, then you can move around the area to see whether you show the plus copy, all right? And then you can release it and then you will drop it. If you find nowhere under this area to show you the plus copy, then what you can do is, what I do is you shut down, you close the Shine tool, all right, and then restart it again.
Okay, I'm gonna try that. Thank you.
Okay, good. please raise your hand if you have done the 708 meta text file and also drop all the sequence file. Okay, it seems most of you have not reached to this step. Okay, it's fine. I give you another 5 minutes. Okay. Are we all in that step now?
Professor, I've actually completed it.
What? What did you say?
No, I have done all the steps. I have also like copied the 708 meta text.
Very good. Okay. So now I want you to open the 708 meta under the Excel. So what you need to do is, okay, I'll show you. Okay, open the Excel, then open the 708 meta, okay, under your folder, all right? Your local folder, not from the China tool, from your local folder, you open. So you will add all these, then you don't need to worry about this, you just click finish, all right? Click finish, then I open the file. So we have H1, H2, H3, H4, 4 different samples, all right? 4 different samples in this metadata file. So you can now look carefully. So basically, what you need to see is this is a file path for the forward sequence, reverse sequence, and then these are just barcode, right? Barcode. So you don't need to worry about this. This is just a location, all right? And here, what you need to see is make sure this is a PWD /708, so that tell that this will tell your program the location of the file is under the folder 708, which we just create. All right. If the folder change, the name change, then these parameters should be changed as well, because this is just to tell us the program, the location of your file, and each of these. So this is the file name. All right. So this is the file name, and this is the folder. Okay. Because we are doing the pair n. So the pair n, so we have the forward sequence and reverse sequence. All right. And for this analysis, it's a 2 times 150 base pair. So that means 150 base pair from the front go to the end. So our primer is 515 to 806. All right. So 150. So that means from 515 plus 150. Okay, that's the 8 region they will sequence. And then the reverse. All right. So since it's 515 to 806, all right, remember that the primer location. So the reverse sequence will sequence from 806 count to 150 base pair backward. Okay, so that's the 2 different sequence location. All right. So this is a brief introduction of the metadata. Once this is done, okay. So now we can start the analysis. Okay. To start the analysis, let me see. You have to go to the open the file that did I give you the 708 script?
I believe it's in there.
It's there. I don't have that as file. Just let me see. Where's the 708 file?
There's a 708 chime script, but it's a doc.
Oh yeah, this one. So the chime script.cocx. I want you to open this file. So you have the file, open that Word file, okay? And then go to the terminal to start the analysis, okay? What I want you to do, very simple. Under the desktop, okay? First of all, you need to demultiplex the sequence, all right? Demultiplex the sequence. To demultiplex the sequence, all right? So what you need to do is you highlight this. This is free. I already wrote it. You click copy, all right? Copy, all right? And then go to the terminal and then paste. Oh, sorry. You cannot just directly because here the metadata file. So the metadata file. Remember the metadata file is 708 meta.txt. All right. So it's a 708 underscore meta.txt. So here you change it to 708 meta underscore meta.txt. So now you copy and paste here. All right. And then they will perform this demultiplex. And it should be very quick. But since I already done this, they probably are confused that I have the file. But it's okay. They still finished. Did you get this green line here? So that produce this file because this file, so the type, this is standard format. So you have Chime tool import, and so I explain from here. So this is the sample data, and there's pair end sequence with quality, all right, and the input pass. So that tell the software to go to the this metadata file, all right. And then since I just showed you the metadata file, we have the file pass, all right. So they will go there, and then they will go through each pass, and then output pass will be. So we assign this file name. So this QZA is artifact. So after the demultiplex, then they give you this parent demux.qza. And this is just the input format, right? And once you go through this, then they will produce this file, the parent demux.qza. All right. All right, so that's the first step, that you will create this file. Once you create this file, all right, then you can try to visualize the file. So to visualize the file, you can just highlight this. First of all, you have to convert the QZA. QZA file to QZV file so that you'll be able to visualize it. So you copy these, all right? And then paste it here. And then they will convert this parent marks QZA to QZV, all right? Then just wait for the software to process it.
Professor,
Yeah.
my one says that like there is a problem importing 708 meta.txt. Like it's not doing anything after that.
The 708 underscore meta.txt is under the desktop. If you go back there, did you see this file there?
Yes, I have copied it to the desktop of the Chime 2 core, and like and then at the terminal I'm copying and putting those commands, but it says that there was a problem importing 708 meta.txt.
So they say they have problem to import this.
Yes.
I have the same file as you do. I have this file, this file. Then the problem would be here.
Okay.
Are this file look the same as mine?
No, I think you haven't unzipped those files, but I have like unzipped and then dragged them and copied them. Should I like
Then
put the zipped ones?
yeah, okay. You go back one step and then put it back here again. Okay.
Okay, Professor.
And then so here you convert this QZA file to the QZV, and now you can try to visualize to see the file. So so the command is trying towards view, all right, and then the file name. All right, so try towards view. So this is a command to view the QZV file. Then you put the QZV file and you click. Then that will open up another window. So now you can see the sequence information. So the minimum is 487 read. The mean is 83,000 read. All right, and then you go to interactive quality plot. Quality plot here. You click here. Now you can see the quality plot, the forward read. All right. So from the first sequence to the 150 sequence, so the quality score normally should be above 25. That give you a good quality. So that tell you forward read until this location 130, 135, very good quality, but you can see at the end the quality get worse. All right, this is very normal when the polymerase they perform this the reading, the duplication, then they lose the quality when they go a longer sequence. I should see the reverse as well. See, if I can see the reverse here, all right. See, this is reverse read, okay, and the quality of reverse read, the reverse read is worse, all right. But since remember we read the 150 both from both ends, and then the total size of the sequence is 292. We have an overlap in the middle, like a 40 nucleotide overlap. So it is okay that in this region and this region is a less the quality is poor, but it's fine. They can still accurately assign the sequence. And here, just to show the percentile quality score, right? So these are the sequence quality information, all right? And now you go back here, and then you click the Q to quit, all right? So you stop and go back to the terminal. All right, so that's the multiplex. And then, of course, that you can filter the sequence, all right? If you want, you can perform this step, that the Q score. So you can have the Q score that you can assign the Q score, like you can set the parameter, and then to perform the Q score, just to remove those bad Q score sequence. And then the next step is called denoise, all right? Denoise. This process, okay, under my computer would take 2-3 hours. So I would not ask you to do this, but I already performed this, okay? So later on, I will send this sequence. These are 3 files. These 3 files. I will send these 3 files to the Blackboard so that you can have these 3 files. Or you can do it after class. Okay. Oh, so I think maybe you should do this. Okay. So I will not give you the file. So you should do this step. Okay. This denoise step. All right. Denoise step. So basically, denoise step is here. All right. Just to denoise, replicate the pair and the sequence. Okay. That's a denoise step. And so the input will be the demultiply sequence that we just did, all right, demultiply sequence. So as you can see here, the sequence here. So we have this pair and demux, this sequence, all right. Use the QZA file. It's here, that's input, all right. So that's the multiplex sequence. And then you have these different parameter, all right, different parameter. So this is the maximum error allowed for the forward sequence, reverse sequence, 10, 10. Of course, this is here, maximum with the expected error, all right? Of if you want to more stringent, so you want to more strict to have less error, then you can set, they have the default is 2, all right? So the number lower, that means it's more stringent, okay, stringent, all right? More stringent, then you get more precise sequence. But normally I would set it 10, all right, that's okay. And then the truncation length for the forward and reverse, all right, forward and reverse truncation, all right. This is truncation length. I mean, the position at which forward read sequence should be truncated due to the decrease in quality. All right, decrease in the quality. So normally I say I will keep it 0, because 0, that means no need to truncate. But from those, when we look at pair and the multiplex, so you can see the forward maybe at the 145 is a better sequence. You can truncate at the 145. And then for the reverse, you can look at the map from these tools view. You can see the back quality at which location. Then you can put in location here. So you can do 120. So that means sequence after 120, they will chop it off. They don't keep it. All right. So that's what truncation means. And then of course they have different parameters that you can explore. Okay. And this one is very important. Okay. So the thread. So if you have a small memory, if your memory cannot run this, all right? If they say if you run this and then after 2-3 hours return a parameter, say okay, we cannot proceed. And then you should add this one, and you can give a number, all right? So so basically the default is 1, all right? So that means the number of threads is 1. And then if it doesn't work, you put a 0. Try to see what happened, okay? So you can put a 0 or back to a 2 and 3 and 4 or 1000, all right? So you can change this parameter number just to make sure your computer will be able to process these command, because this command will use a lot of memory, all right? So it would take time, 2-3 hours. Any question?
No, not right now.
Okay, good. So I think that today we will stop here, and this is what you need to do, all right? This step. I want you to do this step, okay?
Okay.
Okay.
This one, remember, if you use the memory, the RAM, if you my laptop have, because of this trying to I increase my RAM to a 32 it's 32 megabyte, and then as you see that I increase the maximum memory to allocate for China, I use a 24 megabyte. So if your laptop does not have this much memory, that's fine. You can play with this thread, all right? So that they can run it slowly, but will not crash your computer, all right? And then that could take hours, could be 8 hours or 10 hours to run this step, okay? So this is what you need to do this today or this week to get these 3 files. In the end, this output file. So these 3 files very important. So this stat that will give you the statistic of the sequence quality and each sequence and how they define the sequence. And the rep-6, this one is important because this one, you will use this one to run the taxonomy classification. And the table, this one, basically the sequence frequency. So once you get this taxonomy classification, then you combine these and these, then you can perform the alpha diversity, beta diversity, correlation analysis, all kind of analysis. So that's why this is the most important step, okay, at this stage. So I want you to create these 3 different files. Any questions so far? And so I want you to do this twice. One is these 2 maximum allowed error is 10 nucleotide, 10 nucleotide. And then I want you to do 5 and 5, all right? So we can have a comparison. You do 5 and 5. And also another one is 10 and 10. Okay, did you all follow? So you need to do this command twice. The first time you do allowed maximum error 10 and 10. And then once you finish, you do the 5 and 5. All right, so you get 2 sets of data. Any question?
Like, where do you have to put? Do you have to copy the whole thing, from Chime to
No.
stats QZA?
Yeah, from here, data
Okay.
to the noise until the end here.
Okay, and just change the 10 to 5.
Yeah, the first time you do 10, all
Okay.
right, and get the three files. And when you change to 5, all right, change to 5, make sure you change the different file name here. Otherwise, they will override. Otherwise, they will override your data. So you can, for example, change to 5. You can put 5 here, and then 5 here, and 5.
Okay.
So that you can distinguish the difference, and also you will not override your data. Okay.
Okay, thank you so much.
Good. All right. So this is your homework. If you don't have any question, okay, so I will see you again next week. And make sure you have to process until this command, all right? Everyone should
Okay.
process all the way to this command.
Okay.
Very good.
Sounds good.
Professor, for the homework, should we take a screenshot and send it to you or what?
No. You keep those files. Next week we will continue. If you don't have the file, you cannot proceed. So you don't need to show me. You must to have these files so that you can continue to work next week.
Okay, thank you, Professor.
Yes. So here, for example, here, I try to get more accurate sequence. So I do this 8, and then I truncate the sequence at 145 location position, 130 position. So this is what I try. Yeah, but you don't need to do this. I just want you to do 10, 10, and then 5 and 5, and see what happens. Okay, get 2 sets of data. Then next week when we return, then we will continue from here. Okay.
Professor, one more question.
Yes.
So, while closing the Chime2 core, like there are like 3 options. Which one should we choose? Save the machine state, power off the machine, or send the shutdown signal?
So if you are processing, okay, so let's say, okay, I'll show you. So let's say you want to shut it down. Okay, you click this. Then they have 3 options, right? Let's say, as I mentioned, that these command will take many hours. Okay, so you can leave it so that you can continue to run, or you can save the machine state and okay, and so that means that the machine will continue to run the program. Okay, even you close it. Okay, or when you have nothing running, then you do the power off the machine. All right, or when you are running this command, all right, you let it run and just leave it there. Automatically they will just go to this the save the machine state, and then although they are not running, you don't see it's running, but apparently your computer is running the software. Okay, if you are running, just don't touch, just leave it there. All right, leave it there, and then you come back to activate the software after 10 hours, then to see if they finish this command or not. If they finish, then you should see this green line, so that will tell you they finish. All right, if there's an error, then they will show the red line. Okay.
Okay, thank you.
Good, no problem. All right, any other question?
I have a question. It's not letting me paste the commands.
Sorry, Talia.
Yeah, it wasn't letting me paste the commands. I was following along with what you were explaining, just so I had it.
It doesn't let you copy and paste?
Correct.
So did you go to the setting and then go to the general advanced? Did you change this to bidirectional?
I did.
The clipboard and the drag and drop both should be bidirectional.
Oh, just the drag and drop. Okay, wow.
You should do the clipboard as well, right?
Yeah, I just did that. Thank you.
Then you should be able to copy and paste the command, all right?
Mm-hmm. I'm gonna try it.
Good.
Okay, now it works. Thank you.
Good. You are welcome. All right. So if you don't have any further question, then I'll see you next week. Okay.
Thank you.
Okay, bye.
Okay. Bye. Bye.
Okay, thank you so much.
Thank you.
Bye.